Got it.
Let me do that. Good morning and welcome to the Centers of Excellence in Iron and Heme Disorders virtual seminar series. Today we will be hearing from Dean Tantin. And before I bring Dean on to give his talk, I want to share my screen to just remind you of our website and also opportunities for pilot and feasibility. So I'm going to share my screen right now. You should see the website. Do you see that website?
Yeah.
Yep.
Awesome. This is our CIHD website, and you can go in here and find out all kinds of information, the things that are going on at the Utah Center for Iron and Heme Disorders. If you click on this funding opportunity, the Utah Center for Iron and Heme Disorders has a P&F program. It doesn't have a call until June, but the U24 Center for Excellence in Hematology has a current call for pilot and feasibility applications. So if you go here and click on that link, this is the funding call. It's due this Friday, so hopefully you've been paying attention to the emails that have been sent out to remind you regarding it. It's a great opportunity for funds for people who are going in a new direction, or also for younger investigators who are coming up in the non-malignant hematology field. You can find more details here. You can also look to see individuals who received funding before as they're highlighted, and also on tables here. And there's links to websites down at the bottom. So I hope you'll take a look at that. If this is not an opportunity or the appropriate time for you, there will be another call that happens after this one, probably in August. So keep your eyes peeled for it. The Type B pilot and feasibility applications are up to $75,000. And we also have what are called a Type A grant, where if you have a core of interest, for example, at Utah, if you wanted to use the metabolomics core and you have a project that you need that key data for submitting a grant, you can apply for up to $12,000 worth of core. We call it core credits, but it's money that's deposited at that core for your project that you have applied for. So I hope you'll take a look at that and see if that's something that might be a great opportunity for you. And with that, I'm going to pass the baton to Dean Tantin, who is a member of our CIHD here at Utah. Dean comes to us from the Department of Pathology. And I'm not going to take more time of yours, Dean, because I know what a great talk you give. Thank you for speaking for us, and I'll hand it over to you. Great picture, by the way.
Thank you. Let's see, laser pointer. Can you see the pointer okay?
Yes.
And can you guys all hear me just fine?
Yes.
Yeah.
Okay, well, thank you, Diane, and thank you to the CIHD for hosting a seminar for me. I also had a pilot and feasibility grant from the CIHD, and so I'll show you some of the work that we've done, sort of been jump-started by that grant money, including at the end our new lines of human pluripotent cells that we made using the transgenic core and the Colin McGuire's core, which is, I think it's the, what is it, genetic engineering core, something like that.
Yeah.
Okay, so this is where I live, right here where that tin box is. That's my window right there. I'll show you first the people who did the work. This is a kind of old photograph, but it has the two people most involved. So a lot of what I'll show you was done by Yelena Paravanovic, who is a former postdoc in my lab and who is now at Recursion in data science. And she did a lot of the wet molecular biology and all of the bioinformatic analysis that I'll show you. And a lot of the current work I'll show you has been done by a current postdoc in my lab, Julia Shen. So I'll give you a quick overview of, let me just minimize this, of sort of what my lab does. We have a fairly eclectic group of projects, and you can decide amongst yourselves if that's a feature or a bug. A lot of what we do has to do with pluripotency and early embryonic development. I'll show you a project based on that moves into somatic stem cells, including hematopoietic stem cells. And there's also a project in the laboratory that I won't talk at all about a subgroup of T cells that have stem-like properties that endow them with long life and lineage plasticity. Now, there's a lot of common features between these sort of stem-like cell types, and some important differences as well. But the one I want to note is at the bottom, that they all have poised gene expression states. So they all have groups of genes, and this will be important in a moment, that are silent or expressed at a very low level, and they're held in a chromatin configuration that keeps them that way until they have the right development or activation cues that happen. And consistently, some of the same transcription factors control all of those processes. So we have a diverse group of projects, but some of the transcription factors are in common. And also we have a sort of broad overarching idea that the regulation of mammalian transcription is important in the physiology of cell states. And I hope no one would disagree with me on that. So the punchline first, I'll tell you what I'm going to show you, and then I'll go into mechanism. So many of you are probably familiar with this kind of diagram called a Waddington diagram. And it shows at the top a more undifferentiated cell, in this case, a pluripotent embryonic stem cell. And it's going to differentiate down one particular lineage. And that's defined by progressive narrowing of its lineage plasticity and lineage capabilities. But what happens during normal development, and this is in a variety of situations, is that there's a certain leakiness that's engineered into the system. So for example, if you're making a particular tissue or cell type during development, you usually require some kind of mesenchymal support cells around that tissue or around that cell type that are made at the same time during development, although sometimes they can migrate in from the outside. If you're looking at blood development, for example, you might change your myeloid erythroid ratio at different stages of development or in response to different environmental stimuli, but it's very unlikely you'll stop making erythroid cells entirely and only make myeloid cells, or vice versa. So a certain amount of developmental leakiness is built into the system. And that's important for normal development. But if you think about it from the standpoint of regenerative medicine, if you're going to, give someone an autologous blood cell transfusion of red blood cells, it would make sense to use a in vitro differentiation system that is as narrow as possible or as canalized as possible so that you get as pure a population as possible. Even a 3- or 4-fold improvement might change a $100,000 medicine into a $25,000 medicine, which would be a gigantic improvement. And right now, money is definitely a limiting factor when it comes to regenerative medicine. So what I'm going to show you is a knockout of a transcription factor that I'll talk about in a little while called OCT1. When you do this, you see if you go down the same sort of lineage, there's a lot of attenuation and leakiness, and there's a lot more of these sort of side branches that form. And this is fundamentally different from a lineage-specific master specifier like myogenin or MyoD in the muscle lineage, for example, because OCT1 is not lineage specific. It's expressed in all developmental lineages. And regardless of what lineage we make, neurons, cardiomyocytes, red blood cells, we see the same phenotype. The developmental progression is attenuated, and you see the increased expression and gene expression of alternative developmental lineages. And then near the end of the talk, I'll show you evidence that we think we want to follow up using different kinds of ectopic expression systems, integrated expression and transient expression, that ectopic expression of OCT1 can help force or canalize a particular lineage into a more specific state. So we can actually get rid of some of these side branches, which was an unexpected side effect of some of our research. Okay, so what's an OCT1? OCT1 is a homeodomain-containing transcription regulator. This is a review paper that's not our work, but it shows all the mammalian homeodomain, including the biggest group, which are the Hox genes, which includes the CDX genes in here, as well as some non-canonical homeodomains like NK proteins, NKX, paired domain, limb domain, atypical, and then this group here, which is called the POW domain. And this is the group of transcription factors that I'll talk about. The most famous member is this one right here, which is POW5F1, and that includes OCT4, which is the important master regulator of pluripotency in undifferentiated states, and which is used to reprogram somatic cells to an undifferentiated pluripotent-like state. OCT1, which is a close paralog of OCT4, is right here. There's two of these that branch off OCT1 and OCT2. OCT1 is the widely expressed factor. All the other members of this family are expressed in a very tissue-specific, very lineage-restricted pattern. So only OCT1 is widely expressed. And OCT4, if I go back for a second, OCT4, as I mentioned, is an important master regulator of pluripotency. The way it does that is by positively regulating its own synthesis and the expression of a whole group of other master transcription regulators like NANOG and SOX2 and KLF4 that maintain what's called a pluripotency network. So they all regulate each other in a positive fashion and maintain this network. And that's what most people think about when they associate OCT4 with pluripotency. However, OCT4 has a much larger group of targets, over 1,000 additional targets, that are targets that are at a very low level or not expressed at all in pluripotent cells, but can get induced depending on what lineage you differentiate those cells into, either in vivo or in vitro. So those tend to encode lineage master regulators, typically transcription factors, like some of these, for example. If you went down a mesoderm lineage and then into a muscle or a blood lineage, for example, you would first turn on this T factor or brachyury. You would transiently induce PAX3, and then you might turn on RUNX1, and then you might go down a blood differentiation pathway. And all of those genes are direct OCT4 targets. And if you go down that pathway, they switch from a sort of potency in which they're silent but readily inducible to an activated state. Whereas all the other thousands of genes that are specific to all the other lineages, endoderm, ectoderm, for example, are going to get permanently silenced. So we have many OCT4 targets in the pluripotent state, over 1,000 of them, that are equipotential and expressed at a low level. And depending on what lineage you go into, a very small subset of those genes will be induced, and the remainder of those genes will be permanently silenced, with the caveat that some of these factors are reused in multiple lineages, of course. However, there's a problem with this model, which is that there's a sort of black box in this developmental window, so that when you have implantation and gastrulation, or if you differentiate embryonic stem cells in vitro, you lose OCT4 before the induction of brachyury and well before the induction of any of these guys. And so we postulated that there was some other activity, perhaps a paralog of OCT4 that might bind the same genes, that was important for ferrying those poise genes across this black box so that they could get properly induced if you're lineage specific, or permanently silenced if those genes are specific to alternative lineages. And so we tested that, and we looked at that by analyzing an RNA-seq data set. This is work from Yelena Perovanovic, where we essentially what she did was differentiate embryonic stem cells into first mesoderm, and then at day 10, she starts a muscle induction program. And she looked at OCT4 expression and the expression of all the other POW factors and sort of POW adjacent factors that she could detect in her RNA-seq. And you can see, for example, OCT4 is strongly expressed in embryonic stem cells. It's still expressed by day 2. The message is gone by day 6. Very little protein, I'll show you in a second, also at day 6. There's a little bit of OCT11. There's a tiny bit of some other factors. But the only protein that really fit the bill for something that might get expressed throughout this developmental window and could escort those genes through the black box was OCT1. So this is RNA level. I'll show you some data here. That's at the protein level. This is a different differentiation system. So here we're differentiating with retinoic acid, which makes neuronal cells. And what you see here is that OCT4 is almost entirely lost at the protein level by day 6, but OCT1 is retained. And that placed OCT1, and we also had data, and I added this because I thought it was near and dear to my heart. This comes from the first paper that I was ever a senior author on. So this is work from a graduate student at MIT that I sort of mentored when I was a postdoc. And what she did was make a knockout, a germline knockout of OCT1. And that particular allele, when it was knocked out, which is a severe hypomorph, I'll give you the true null phenotype in a second, resulted at embryonic day 11 and embryonic day 12 at severe runting. That's the most obvious phenotype you can see. There's some hemorrhaging, which you can see better with PCAM staining. There's some heart defects, and there's also this pale fetal liver phenotype that you can see right here. And you can see that a little bit as well using TER119 staining. There's a reduction in TER119. There's probably another developmental block beyond the TER119 stage at enucleation, for example, and hemoglobinization. And it's specific to the red blood cell lineage because GR1 cells, granulocytes, don't have that defect. Additionally, if we take that fetal liver and do a transfer, a bone marrow transplant, we can show that those recipient mice do not efficiently make red blood cells that are derived from that fetal liver. The same year, we published a second paper showing that that fetal liver was in fact capable of giving rise to B and T cells. So there was some cell activity there. But subsequently, we showed in 2012 that if you do serial transplants or competitive transplants, then all of those fetal liver cells crap out. They can't do a serial repopulation, for example. And that manifests at the level of the hematopoietic stem and progenitor cell, the LSK cell, which is the level at which we did it back then. And so I'll also tell you subsequently a more severe null was made by a lab in Switzerland, Patrick Mathias's lab. That mouse has a developmental arrest very early, and it's due to a placental defect due to trophoblast stem cells, which is a phenotype I won't talk about. But if they bypass that defect doing some developmental tricks, they get an arrest a few days earlier than 12.5, and the animals are again runted. There's a heart defect, there's fewer somites, so there's a developmental arrest. And most of what I'm telling you, somites, myocytes, vasculature, fetal liver, those are all mesoderm in origin. And so we had a specific reason to study mesoderm in this system. But nevertheless, it looked like OCT1 was a protein that fit the bill because it was expressed at the right place and the right time to potentially ferry those genes across the black box. So we decided to test that. So way back, I showed you 20-year-old data where we made a germline knockout. More recently, we made a conditional allele, and we've deleted that in a number of adult tissues. We've deleted it in the gut, and we've deleted it in T cells, for example. I won't tell you anything about that. But we also crossed the floxed allele to a Rosa Cree ER. We derived new lines of embryonic stem cells from those mice. And we treated those mice with 4-hydroxytamoxifen to delete OCT1. And you can see the parent cell line has OCT1. 2 different derived lines lack OCT1. And when we put those embryonic stem cells into culture, they looked, I'm not showing you pictures, but they looked normal. We could propagate them indefinitely. They also expressed the components of the pluripotency network. So they expressed OCT4, NANOG, and SOX2. However, they differentiate abnormally. And I'll show you some molecular data, but the most obvious physiological data is if we take those embryonic stem cells and we put them into blastocysts, early embryos, and these are black embryonic stem cells, and we put them into albino blastocysts, the parent cells can contribute to development in those mice, whereas the knockouts do not. So this is an even worse phenotype than the severe hypomorphic allele that I showed you previously. So this indicates that OCT1 deficient embryonic stem cells must differentiate abnormally. So we tested that by bulk RNA-seq. This is data that was done by Julia Shen in my lab and published in this eLife paper back in 2017. If you just look at the parent embryonic stem cells and you compare them to a isogenic derived tamoxifen-treated line, at the embryonic stem cell stage, so in an undifferentiated state, basically all of those dots, which are genes, fall along the diagonal. And that indicates there's very little difference in gene expression associated with OCT1 deficient embryonic stem cells in an undifferentiated state. But all of those red genes that are highlighted here will be significantly differentially expressed when we differentiate after 14 days. So this is retinoic acid differentiation. Those cells are going towards neuronal cells. And you can see a group of genes that are downregulated. That includes a lot of neuronal specific genes like HOXB9 and HOXA5. However, there's an even bigger group of genes that are abnormally upregulated. And those upregulated genes include SOX17, CDX2, PPAR gamma, and prolactin 8A6. SOX17 is ectoderm specific. CDX2 is expressed in the trophectoderm. It's also expressed in early blood cells and in the gut. So those are different lineages than ectoderm. PPAR gamma is expressed in mesoderm cells that are going towards adipocytes. And prolactin 8A6 is specific to the trophectoderm and the placenta. So these are all genes that should not even come up in lineages that you're making from embryonic stem cells. Prolactin 8A6 shouldn't come up in embryonic stem cells generally because they only make tissues of the embryo proper. So there was a severe abnormality that we saw upon differentiation. And it's not a kinetic problem. So you could imagine, well, maybe if you culture these cells for a little bit longer, these genes eventually come up and all there is a delay. And that's not the case because in a downregulated gene, we can see it downregulated across the time course. In an upregulated gene, we can see it upregulated across the time course. So there was something really at the gene expression level phenotypically abnormal in those cells. We could also take those cells and make cardiomyocytes. Those cardiomyocytes fail to beat. We can make true neurons. They fail to arborize. They fail to make beta-3 tubulin. This shows myotubes. So in this case, we're making mesoderm and then myotubes. And you can see the parental cells fuse and make myotubes just fine, but the knockout cells, conditional knockout cells, fail to do so. And that's also at a later time point associated with defects in gene expression of these myogenic master regulators like MyoD and myogenin. So we ran bulk RNA-seq at a much earlier time point as the cells are specifying mesoderm, with the idea that the gene expression defects we identify early might explain some of the later phenotypic defects that we observe. So we did 3 time points, undifferentiated, day 3 and day 6. This is just as OCT4 is getting lost in parental cells and in conditional knockout cells. And we see a number of interesting changes, but the one I want to focus on is this cluster 2, where there's a group of genes that get induced in the parental cells, and they get induced much more poorly in the conditional knockout. And those include a number of genes that are important in specifying mesoderm, like PAX3 and PAX7 and delta-like1. We can also make, and this is work that's funded by the CIHD, this is unpublished, we can also make blood cells out of the embryonic stem cells by first specifying mesoderm and then specifying blood lineage cells. Both primitive and definitive erythrocytes are quantified here by colonies. And the primitive guys, you can see there's a severe defect. We didn't get any definitive colonies at all. If you look at those colonies, though, they look somewhat normal. There's some hemoglobinization happening. You can see the pink tint, I hope, on your screen. So it's sort of a numbers game. The cells that we get seem to be phenotypically normal. If I back up, the few that are not shown in this field, but the few myotubes that we get seem to look normal in terms of their amount of staining. It's just a digital readout where you get fewer of the correct cells. And we've also done this more recently by flow cytometry. So if you look at BFUE and CFUE, based on the parent and the knockout, this is done at an earlier time point than what I showed you before. We have, and this is, I would say, not very stingy gating. So we're being very generous in our gating here. If we're stingier and we move the gate, we can probably see a bigger fold difference. But at this time point, just at day 6, we can see a reduction in colony forming units, erythroid colony forming units. So at this point, we decided to look at with more granularity at the populations of cells that we were getting. So we switched to single cell RNA-seq. This was published back in 2023 in a Science Signaling paper. And at day 6, which is what we're looking at here, you can see some cells that retain an embryonic stem cell-like feature, some cells that retain an epithelial state, a number of cells here that are hard to characterize that we just call oxidative stress because of some of the genes they're inducing. And then all of these clusters of cells here that are moving towards mesoderm, paraxial mesoderm, and then somites and blood progenitors. So the cells generally progress in a sort of shape that looks like this based on pseudotime. And this sort of developmental leakiness is part and parcel of what I talked about at the beginning, where you do have a certain amount of imprecision in these cultures and also in the developing embryo. We can also look at individual clusters. This is the neuromesodermal cluster, and you can see they've downregulated a number of important genes for mesoderm specification, and they've upregulated a bunch of epithelial genes, which is abnormal. That's inappropriate, and that happens in multiple mesoderm clusters that we look at, including the blood progenitor cluster. We can run pseudotime on our single cell RNA-seq. So what you do here is you specify one cluster as a beginning. And I think we use the neuromesodermal cluster as the endpoint that goes to both of these clusters towards the end. And when we do that and we overlay our single cell RNA-seq at day 3, at day 0, day 3, and day 6, in the parental cells, you can see most of the embryonic stem cells cluster up at the beginning of the pseudotime. The neuromesodermal cells cluster at the end. There's 2 developmental branches that are sort of developmentally inappropriate that happen in this cell type. However, in the conditional knockout, you see a lot of the cells still cluster at the embryonic stem cell state in the day 0 condition, but many fewer cells make it to the neuromesodermal population. And the abnormal branching is much more pronounced in the conditional knockout situation. And we think this is conflating, based on the way pseudotime works, a number of inappropriate developmental branches at the same time. And we call them epithelial cells to be simpler. As you know, mesoderm cells have to undergo a epithelial mesenchymal transition in order to specify mesoderm. And that's induced by TGF-beta, which is going to be important in a moment. We can look at individual genes through pseudotime. So we're looking at KLF4, which is a pluripotency-associated gene, T or brachyury, which is transiently induced to specify mesoderm, and then PAX7 and CXCR4, which are they're at the end of this time course, but they're induced during the mesoderm specification program. And so you see KLF4 strongly expressed at the beginning, it's lost, TBXT transiently or T transiently induced, and then PAX7 and CXCR4 induced towards the end of that process. What happens in the conditional knockout cells that have been treated with tamoxifen is that KLF4 is abnormally retained for a little bit longer. I would say TBXT or brachyury gets induced normally, but it abnormally lingers. So you have a problem resolving brachyury expression. And we have very poor induction of both PAX7 and CXCR4. So based on this anecdotal analysis, Yelena in this study did what we call developmental coherence analysis. So in this analysis, what she did is she took the 1,000 most dynamically expressed genes over pseudotime, and she clustered them together. And you see there's a group of genes that are associated, and I don't know how to get rid of the panel, but I can basically guesstimate what the genes are on the right-hand side of my screen. On the left-hand side here, you have a bunch of genes that are expressed at the beginning of pseudotime, which are associated with pluripotency, such as PAL5F1 that encodes OCT4 and NANOG and KLF4. You have transient induction of genes like TBXT or brachyury. And then you have genes that are expressed in a very tight wave progressively, like PAX7 and PAX3, for example, until you get to genes that are associated with mesoderm specification and terminal differentiation. And I have PAX3 and PAX7 down here, which I can see. So there's stuff up here that I can't see that's associated with that middle step. And I'm sorry, I can't read that with my laser pointer on. In the conditional knockout, you start out pretty normally. So you start out expressing NANOG and OCT4 and the pluripotency-associated genes. You have a defect in the developmental specifiers at the end, which I showed you and which we expect phenotypically. And what's interesting here is you have a loss of developmental and temporal coherence in the induction of these transiently expressed genes throughout pseudotime. So brachyury is sort of spread out in its expression. These genes lose developmental coherence, so they're no longer synchronized properly with each other. And that's just consistent with the sort of fact that these cultures are going off in all kinds of crazy directions. So at this point, we can find that OCT1 is expressed with OCT4 in the pluripotent condition. It's expressed throughout this black box period, and it continues to be expressed. And if you take it out, we know these genes are being abnormally induced. And so the question then is, what is the mechanism? What's happening during that black box period that OCT1 is doing that's allowing these cells to properly canalize into the right developmental specificity? So in order to introduce that, I'll give you a little bit of background. In the embryonic stem cell state, when you have OCT4, then we know now OCT1 is co-expressed with OCT4. We know from ChIP-seq results that have been done by others that OCT4 binds a number of important developmental regulators. Those regulators are associated with a particular chromatin state called bivalency, which is associated with the simultaneous presence of activating marks, such as H3K4 trimethyl, and repressive marks, such as H3K27 trimethyl. It's also associated with a lack of DNA methylation. And that's important for keeping these genes in a poised but readily inducible state. And the question that we had was, where does OCT1 fit in this picture? And so to do that, we did ChIP-seq analysis of both OCT4 and OCT1, looking at OCT4 during the embryonic state and looking at OCT1 in embryonic stem cells and then during development. So if we just focus on the embryonic stem cell-like state, hopefully you can appreciate with this Venn diagram of OCT4 and OCT1 targets that many of the OCT4 targets are also co-bound by OCT1 at the same time. And we can use rechip or sequential chip to show that actually these two factors are simultaneously bound to these targets rather than being bound in an alternating fashion. And how is that possible given that you have a binding site? I don't have time to get into that, but if people are interested in the question period, they can ask me. It has to do with the nature of the binding sites for OCT1 and OCT4. So we have many genes that are co-bound by OCT1 and OCT4 at these developmental specific genes. And in fact, if we look at, in this case, day 6 of differentiation and OCT4 is no longer expressed, but we can do OCT1 ChIP-seq, what we find is that about 10% or 8% or so of those genes continue to be occupied by OCT1 during mesoderm specification. And if we look at what those genes are, they're important in notch signaling, TGF-beta signaling, as well as mitochondrial reorganization. Metabolic changes are important in mesoderm specification and oxidative stress. So these are the right kinds of genes that are associated with mesoderm specification patterns. So that suggested to us that OCT1 and OCT4 are co-bound in the bivalent state, in the pluripotent state, that where all of these lineage-specific genes are equally inducible, and that OCT1 continues to be bound to those genes that are going to get induced. You can ignore the stuff on the right side here for the moment. The question then is, well, the lineage-specific genes are in the same genome as the lineage inappropriate genes. What is it that's different about these genes and not present in this genes that allows OCT1 to perhaps remove those inhibitory marks and allow those bivalent genes to resolve and get induced properly? Actually, before I talk about that, I want to talk a little bit about the mechanism. So we postulated that there was a factor that was removing this epigenetic mark that was based on previous work we'd done in the lab, indicating that one activity of these factors, one way they regulate gene expression, is by recruiting histone lysine demethylases, and these are methylated lysines. So we looked at the activities that we know from the literature can remove H3K27 trimethyl marks. And the obvious one to look at was ultratrithorax, because that was associated with defects in mesoderm when it was knocked out. So we did ChIP-seq on OCT1 and on ultratrithorax. And surprisingly, I was hoping that we had like 5% of the same genes, but really surprisingly, these are peaks, not genes, but about 4,000 of the peaks that were, or about half of the peaks that were associated with ultratrithorax in cells differentiating towards mesoderm were co-bound by OCT1 in those same conditions. And that indicates a very strong association between OCT1 and ultratrithorax for binding. And we could also then subsequently show that ultratrithorax is expressed throughout the differentiation time course, and that it interacts, this is covered up, but I'm assuming there's a band there, that it interacts in co-IP with OCT1. So at this point, we can place ultratrithorax in that position. And then the question became, well, why is OCT1 recruiting ultratrithorax over here and not over here? And to address that, we went back to our ChIP-seq data, where we could use, this is now a ChIP-seq track at day 0 with OCT4, day 0 with OCT1, day 6, and day 6 with ultratrithorax. And we can see ultratrithorax binding to enhancer elements, not necessarily at the promoter. But down here at the promoter, we could see SMAD3, which is an activity that acts downstream of TGF-beta signals. That was associated, and this is a site that's computationally predicted, that was associated with known OCT1 peaks. And this is at the promoter area in this particular case. So we did a broader analysis of all of the OCT4, OCT1, the intersection of OCT4 and OCT1 peaks that were maintained during mesoderm differentiation, that 800 or so OCT1 peaks that were present. We found the OCT site. The computer is calling it an OCT2 site, but it's an OCT1 site, ATGCAAT. And in addition to that, we also found binding sites for SMAD proteins, the downstream effectors of TGF-beta signaling. And as I mentioned, TGF-beta is a master inducer of mesoderm specification. So we decided to look at whether or not SMAD2 was also present at some of these genes. We found, and I'll skip this in the interest of time, that OCT1 and SMAD3 interact, and that was actually a replicate of data that was published previously by other groups. And we showed that OCT1 and SMAD3 display cooperativity in reporter assays. These are old school assays that I used to do as a postdoc. And we went back to some of those assays. And it turns out that if you take these binding sites ex vivo and you put them into cell lines, in this case fibroblasts, you can get synergistic luciferase reporter activity out of co-transfected OCT1 and activating the TGF-beta pathway. So now we can add to this diagram, we can add SMAD3 to that diagram and basically have a model in which when you have lineage specifiers for mesoderm at the specific subset of genes that are necessary to be induced, there's a cooperation with OCT1 that leads to the recruitment of ultratrithorax, the removal, among other things, the removal of this inhibitory mark, allowing these equally poised genes that are specific to the mesoderm lineage to get induced during that developmental pathway. So what was left here, which I'll go over relatively quickly, was what's leading this repression. We already knew that in repressed genes, OCT1 interacts with this repressive chromatin activity called NERD. We knew from the literature that NERD interacts with LSD1, which is important at enhancer elements in terms of its ability to remove methylated histone marks. And at this point, I'm going to have to do a tiny little razzle-dazzle and a tiny little sleight of hand because H3K27 trimethyl, the mark that's removed is a positive mark that's removed by LSD1. And instead of H3K4 trimethyl, which is only present at promoters, the mark that LSD1 removes is H3K4 mono and dimethyl, which is an enhancer-associated mark. So we have a model where LSD1 removes this mark, and then subsequently that's converted into loss of H3K4 trimethyl at the promoter. That's the current model that we have. And to test that, we did ChIP-seq using OCT1, which we already had, NERD and LSD1. We looked at the intersection of OCT1 and NERD binding sites and what genes, what peaks intersected with LSD1 ChIP-seq at this particular stage of development, which I think is day 6. And again, we found binding sites for OCT1, which was good because these are OCT1 binding sites. In this case, they call it POW2F3, which is OCT11, but it's the same site. Except now we saw many weaker motifs, and those included things like FOXA2, SOX2, SOX17, CDX2, and HNF4, which are associated with endoderm, ectoderm, and endoderm. So now we have, in these repressed guys, lineage master regulators for alternative lineages. And that leads us to a model where if you have the presence of lineage-specific master regulators, so if you're making mesoderm, you have the presence, you have TGF-beta signals and the presence of SMAD3, you can recruit ultratrithorax and you positively resolve bivalency. In those mesoderm-specifying guys, you have other genes with binding sites for FOXA1, SOX2, and SOX17, CDX2, HNF. Those guys are not present by and large. Some of these factors get recycled, but by and large, they're not present in cell-specifying mesoderm. And so the idea is that OCT1's default transcription output is repression. And in that case, the enhancers get nullified by removing through LSD1, H3K4 mono and dimethyl, and that gets converted into loss of H3K4 trimethyl at the promoter and permanent silencing, addition of DNA methylation of those genes. So that's the mechanism. And I think at this point, we have relatively good data that we can now place OCT1 into this developmental trajectory, including across the black box, so that it can ferry these genes across so that they can get properly induced or more permanently silenced. And hopefully I've provided a molecular mechanism that can explain that. So if I was just to summarize what we have here, OCT4 hands off its functions to OCT1 as pluripotent cells differentiate and OCT4 is lost. OCT1 deficient pluripotent stem cells have a defect in terms of their ability to canalize into mesoderm. In cells undergoing mesoderm specification, OCT1 cooperates with mesoderm's defining transcription factors such as SMAD3 to recruit chromatin modifying activities such as ultratrithorax that remove inhibitory marks and positively resolve bivalency. And in the absence of those cooperating master regulators, OCT1's default mechanism is repression. Any questions about that so far? Okay, so the last little sort of quarter of the talk or so, I'll talk about a new direction that we're writing into an R01 at the moment and hoping to get funded. And this basically stemmed from work that Yelena, a former postdoc in my laboratory, did, where she was looking just to make sure that there was nothing weird about these conditional knockouts that were tamoxifen treated that was causing a developmental defect, and that it was actually due to OCT1. So in order to do that, she complemented the OCT1 loss by retrovirally transducing these cells with OCT1, and then made muscle cells. So she takes these out to day 11 and does muscle cell specification in vitro. And so these are the red cells. They've been normalized to the parent cells just so that you can see what I'm about to tell you more easily. But what you can see is that when you differentiate towards mesoderm, you have an increase in those OCT1 deficient cells and their ability to make MyoD and myogenin. And you have a decrease in this activity here called PAX3, which is transiently induced during the mesoderm specification program in order to make muscle later. And PAX3 is downregulated. So the combination of PAX3 being downregulated and these myogenic genes being upregulated indicates that the cells are more efficiently transiting through a mesoderm specification program and making muscle at the end. But what was really fascinating here was that the parental cells were also doing the same thing. So in cells that already had OCT1, if we ectopically expressed it, even in a crude constitutive way using a lentivirus, we would have higher expression of MyoD, higher expression of myogenin, and lower expression of PAX3. And that would be consistent with a model in which OCT1 could also improve myotube generation. So that suggests a potential practical application in regenerative medicine, improved canalization. So a postdoc in my laboratory, Julia Shen, tested this by making myotubes in culture. This is day 18 of culture, so this is a suboptimal time point and slightly suboptimal conditions. You can see she can generate some myotubes here with the parental cells. They're GFP transduced just so that we can see the cells more easily, and they've been transduced with an empty vector. These guys are also GFP positive, but now they're transduced with OCT1, so they're ectopically expressing OCT1, and you can see they make a lot of myotubes. These are basically just replicate cultures that are shown here, and the quantification is shown on the right. So we actually tried some initial experiments where we damaged muscle and we tried to engraft cells into the muscle and did reclosure surgery and then open them back up. We could see cells engrafting, but we didn't get much further than that, partly because Yelena in my lab had taken a job at Recursion, but also because we realized that blood cell differentiation was the much more efficient way to go about doing this. It's easier to make blood cells. You can engraft them into animals more easily and study effects in vivo. And more importantly, some of the first regenerative medicine that looks likely to be deployed going forward in the next 5 or 10 years is likely to be blood cell in origin. There's over 10,000 allogenic transplants that happen in this country every single year. And that's associated with difficulty in finding matched donors. Even a 5 out of 6, so a poor match, is sometimes used because the donors aren't matched. You can't find good matched donors. And that's often associated with all kinds of comorbidities, such as graft versus host disease. So in some of those patients, if you could basically do autologous transplants instead by taking skin cells, for example, from that patient, inducing pluripotency, and then in culture, making red blood cells, for example, putting them back into the patient, that would be a much more efficient way of going about doing this than the autologous transplants that are currently standard of care. So we looked, again, using the same methodologies I showed you earlier, using erythroid colony formation. And we found using ectopic expression of OCT1 that we had higher levels of both blast forming and colony forming units. We're currently doing this by flow cytometry, but that data is not there yet. And we're currently doing in vivo engraftments, and those mice are ordered and about to get engrafted in the laboratory. But rather than sort of go down that rabbit hole of mouse engraftment and mouse culture, we decided to pivot because it turns out the hematopoietic differentiation field has really matured in the last 10 years or so to a point where doing this in human cells is actually easier than doing it in mice. And in vivo, you can use immunocompromised mice and you can look at human blood cell engraftment. So using CIHD funds, we used our core facilities to generate knockouts, OCT1 knockouts of human embryonic stem cells. We did this using transfected CRISPR-Cas9, and this just shows the schematic right here. I'm skipping over the genotyping and the HRMA. It's not very interesting, but I'm showing you a Western blot of these embryonic stem cells that shows they've efficiently deleted OCT1. And exactly as what we saw in mouse cells, when we delete OCT1 and we look at those cultures, they look normal in terms of their developmental status. They look like they're undifferentiated pluripotent cells. They still express OCT4. We haven't looked at SOX2, NANOG, KLF4, and we haven't differentiated these cells in culture yet. But basically what we're gearing up to do is a complete analysis of blood cell development in OCT1 deficient human ES cells, make sure we have the same kinds of deficiencies, and we'll take the parent cells and either permanently or transiently ectopically express OCT1 during the differentiation program to see if we can get better blood cell differentiation in vitro and in vivo out of those cells. And so that's the current, and we'll probably use iPSCs, not human ESCs, because these are too high passage for those experiments. So that's where that work is going at the moment. And so if I was just to sum up in a single slide, everything I showed you, we now have a mechanism where we think OCT1 ferries lineage specifying equipotential genes out of pluripotency and into a state where they are stably expressed by recruiting factors that remove these inhibitory marks, or permanently silenced by recruiting activities that remove these activating marks. And as a consequence of that, when we knock out this factor OCT1, we find that the cells have a strong failure to canalize in terms of their differentiation status. And we have emerging data that when we ectopically express OCT1, we can get superior differentiation outcomes, which might have implications for regenerative medicine. So let's see, 9:50 or so. I'll stop there. I'll just acknowledge the people that worked on this, and then I'll take any questions. So this is my current lab. I should have highlighted her, but Julia Shen is the one who is doing the work currently in the laboratory with flow cytometry, colony counts, and the mouse engraftments, looking at blood cell differentiation. A lot of the early work that I showed you, including the muscle work, was done by Yelena, who was a former postdoc in my lab, who is now at Recursion. And we've been helped by a number of other former and present members of the laboratory. Here in Utah, which is where our collaborators have been for this project, I want to point out the high throughput sequencing core, as well as, not shown here, the transgenic core and the stem cell core run by Crystal and run by Colin. A lot of the cloning and reporter assays that we put together, we were helped by Mahesh Chandrasekaran, who's in radiation oncology here. We did a whole series of experiments on topological interactions between binding sites for OCT4 at enhancers and promoters. And that isn't work that I showed you in the interest of time, but that work basically indicated that OCT1 piggybacks on these topological nodes of interactions to help regulate gene expression. And that was work that we did in collaboration with Dr. Jake Gertz in the Department of Oncological Sciences. And then some of the hematopoietic differentiation stuff, the flow cytometry, we've been helped by Hans Hecker's lab, who is in my department, pathology, and a student in his lab, Faraz Salahi. So some of the open questions: can ectopic OCT1 improve blood cell differentiation in vivo, in vitro, either by integrating OCT1 or more likely to be used for regenerative medicine, transient expression of OCT1 using Sende virus or adeno-associated virus, for example? Is there an optimal window during differentiation when OCT1 needs to be expressed? And can we recapitulate those findings using new human stem cells that we've just generated through funds that we got from the pilot and feasibility grant that we got from the CIHD? I also want to point out these funding sources, and I'm happy to take any questions anybody has. Thanks for your attention.
Thanks, Dean. Great talk. I swear I learned so much every time I listen to you talk. I'm going to start with a question myself.
Sure.
In your hypomorph, did you have is there an optimum level of OCT1, or do you see anything in a heterozygote? Like not sufficient levels of OCT1?
Right. So you're going back to the 20-year-old work on
Yeah.
those hypomorphic mice. So those mice are really severely hypomorphic,
Okay.
right? So if you just do a Western blot, you can't detect OCT1. But in that paper, what I did is I used affinity purification. People were much more biochemical back in those days. And I enriched for OCT1, and I could actually see a tiny bit of OCT4 degradation products that were present, sorry, OCT1 degradation products that were present in fibroblasts that were made from those same embryos that showed the pale fetal liver. So there's a tiny, tiny bit of OCT1, which we showed in that paper. What I take out of that result, which is, I think, very important, is that the amount matters.
Yeah.
And it matters a lot. So when we say we've ectopically expressed OCT1 using a lentivirus, that is very crude. And we have no idea what degree of expression. I mean, we know it's overexpressed, but we have no idea what degree or what timing is important.
Okay.
And that's because a complete knockout, a null, has a much more severe phenotype where you have a placental defect, where you see no placental defect. I mean, there might be some reduced numbers in terms of litter size, but there are definitely mice that can make it past that developmental restriction. And so the amount of OCT1 is very tightly associated with which developmental restrictions you can get through. Does that answer your question, or did you have something?
Yes, no, that's exactly what I was sort of expecting, is that there's sort of an optimum level. And so you're talking about using OCT1 overexpression potentially as a sort of regenerative medicine.
Right. I mean,
tool.
what's
And so
really
I was trying to
crazy,
figure out what's the level.
yeah. So what's really crazy is that's work that I'd forgotten about, a decade ago. And here we are 20 years later, and we have new data that suggests this whole thing is going full circle, and we're sort of pivoting back, and we're actually reread that paper recently. So it's just one of those things where science, good science, I hope, but lots of different kinds of science often cycle back and build on things that you sort of had just passed over and moved on to bigger and better things. But it turns out there was something interesting there that we're now going and following up.
Well, Phil would be proud, right?
Yep. I'll tell him that I'm going there in June. So.
Oh, good. Okay. Awesome. Other questions? You feel free to unmute yourself and ask. I don't see anything populated in the chat.
I have a question.
Do it, Mark.
Yeah.
Okay. Thanks, Dean. That was great talk. Similar to Diane, I also learned a lot. I had a question about so when people do these like stem cell therapies, has anyone ever screened these cells for OCT1? Are there different levels of OCT1 in the current therapies being used? And does that guide it towards, mesodermal or ectodermal or whatever fate? And then also with your regards to your research, is the end goal, do you think, to do like a genetic therapy on these transplanted cells for OCT1?
Right. So to your first question, I don't know. I suspect that a lot of the clinical research on stem cell therapies has been sort of phenotypic in style, where you're basically trying to use culture conditions that are approved, FDA approved, and also that give you maximum output. But some of those studies might be associated with some genomic analysis that we might be able to go back and reinterrogate and see what the expression level of OCT1 is in some of those cells. So that's a good idea. It's something we should go and look at for sure. Your second question, no, I don't think our end goal is gene therapy or anything that integrates or modifies the genome, because best I know, there's nobody walking around with deficiencies in OCT1, because those people are by and large never getting born. There are some polymorphisms associated with OCT1. I don't know if there's diseases associated with that, though. Instead, I think the idea is that if you have patient-derived pluripotent cells, and you're going to use that, let's say that patient gets into, I'll move out of blood cells just for a little while. Let's say that patient is in a car accident and they have some kind of spinal cord injury and they're paralyzed, right? You want to take their stem cells and make not just neurons, you need to make motor neurons, and not just motor neurons, you need to make cervical motor neurons, not just cervical motor neurons, you have to make C3 cervical motor neurons. And so when you do that, even with the optimal conditions where you have all the right growth factors and the right cookbook to make those motor neurons, there's still a very tiny fraction of your culture just because the culture is very dirty and you have to purify those out, and there's all kinds of things associated with that. What if there was a factor that helped canalize differentiation such that if the right inducers are present, you made 10 times more of those neurons, even if it's just a transient association, nothing that permanently modifies the genome, by adding this OCT4 paralog OCT1 at the right developmental step. That's the idea. Whether this works in non-mesoderm lineage cells, we don't know. We've just focused on mesoderm because of the knockout phenotype. But we think in blood cells, this would be something that we could actually apply and get better differentiation outcomes, maybe muscle as well.
Super
It'd be
cool.
fun
Thank
if
you
you
so
could
much.
treat aplastic anemia.
Yeah, yeah.
Yeah,
So erythroid, it looks like we're getting a big erythroid phenotype out of this. Yeah.
yeah, that could be kind of cool. Because I mean, you either self-resolve, or I'm not a clinician, so I don't know the details, but you either self-resolve and they don't know why, or you succumb to it.
Mm-hmm.
So that might be a cool sort of red cell lineage analysis that could be done. Any other questions? As I know, Dean, great talk. Thank you. I've learned
Thank you.
so much. And thanks everybody else for dropping in. Hope to see you next month when Tracy, who's gone off the Zoom, will be our presenter, and she's going to talk on iron sulfur cluster proteins in mammalian cells and viral infections, which is kind of fun.
Okay, looking forward to it.
That's
Okay.
good. Great talk.
Thanks,
Dean.
everybody. See
Really
you.
good.
Good seeing
Thank
you.
you.
See you.
Bye.